In this module, we are going to talk about something slightly different. It is, since we are on the subject of DNA replication and we are talking about DNA, it is important that we talk about some other technologies that also exploit special features of DNA. We are going to talk about restriction endonucleases. These are enzymes that can cleave DNA at specific sites. I would like to say a few words about their discovery. It is important. The scientists noted that bacteriophage, the virus that can infect a bacterial cell, a bacteriophage that was infecting E. coli, a strain of E. coli, B strain, when this virus was used to infect a different strain of E. coli, it was a E. coli K strain, it was very ineffective in infecting that E. coli K strain. After several generations, the virus was adapted to infect and grow in the E. coli K strain. When the same virus, which was originally taken from the E. coli B strain and was then grown in E. coli K strain, initially it was difficult, but later on, when the bacteriophage strain adapted to E. coli K, they took that bacteriophage and tried to infect E. coli B again. So here, bacteriophage that could grow in E. coli B was taken to E. coli K. Here, it was not able to infect this K very well. However, after several generations, it was, and then same virus was used to infect E. coli B. This virus could not infect E. coli B very efficiently now. So this puzzled the scientists and they wanted to look further into it. So let's look at what they found. They found that special bacterial enzymes can cut double-stranded DNA at specific sites in the test tube. It so happens these bacteria, all cells, can modify their DNA, and their chemically modified DNA serves as a protective mechanism. So cells know this is our DNA. Any foreign DNA that tries to enter the cell which does not have those chemical modifications is basically destroyed. So that is why the phage that was initially able to infect the E. coli B very successfully, when it was taken to E. coli K, it was not able to infect E. coli K very successfully because it was being degraded. Their bacteria also have a sort of an immune system that detects foreign DNA and degrades it. However, after several generations, that bacteriophage DNA also accumulated same chemical modifications and was able to infect E. coli K very successfully. Now, that bacteriophage in E. coli K strain had a different type of chemical modifications. When it was taken back to E. coli B strain, it could not infect the E. coli B strain very well. That is what led scientists to explore this phenomena, and they found that there are enzymes, bacterial enzymes, that can cut double-stranded DNA at specific sites. Since these enzymes restricted the growth of bacteriophage, they were called restriction enzymes. They restricted the growth of a phage virus. These viruses act as sort of immune system of bacteria protecting the invasion of foreign DNA or the viruses. Many recombinant technologies which have been invented in the field of biotechnology heavily rely on the development of or the discovery of restriction enzymes. So it's very important. I would like to point out a few things about these restriction enzymes, some of them which we are interested in. These enzymes cut a palindromic sequence. What is a palindromic sequence? It reads the same whether you read it from 5' to 3' direction or 3' to 5' direction. For example, easy to understand, the word Dennis-Sind. If you read it from left to right, it reads Dennis-Sind. If you read it from right to left, it also reads Dennis-Sind. This is what a palindromic sequence is. These enzymes, restriction endonucleases, they cut DNA at specific locations, which are generally palindromic sequences. And when they cut the DNA at palindromic sequences, some of these enzymes can generate sticky ends. What do I mean by sticky ends? Some of these enzymes cut DNA. They will have single-stranded DNA in which the bases are exposed, and these bases have ability to form hydrogen bonding, which you can see right here. So they can form hydrogen bonding between complementary nucleotides or a fragment of DNA with the complementary nucleotides. So this is basically what I wanted to talk about, restriction endonucleases. We will see how endonucleases can be exploited in the field of biotechnology in a later module.